Thanks, everyone. It's a privilege to be here, and I want to first thank the West Coast Metabolomics Center for the funding support, which allow us to start the path to really answer a long-term study goal of ours. And that study goal is actually to improve treatment for one specific neuropsychiatric illness. The illness is anorexia nervosa, and this is a picture of Rachel Farrak. Some of you might have heard of her because she actually became a self-nominated public figure for anorexia nervosa just during this year when she became really ill, and she posted YouTube videos asking for donations so that she could get treatment, specialized treatment, which unfortunately is not being heavily utilized to treat patients because most of them have a strong denial of their own illness. So this is her 10 years ago when she was young, beautiful, weighed a healthy 125 pounds, and this is her basically this year. She's still at the same height of 5'7", but she's weighing less than 50 pounds. In fact, just last month, she had a couple of ICU crises because of extreme low potassium, and currently she's being treated in the UCLA eating disorder specialty clinic. So do all patients with anorexia look like her at the current stage? No. They usually start out looking healthy. The onset is at the teenage year, and only 2% of the population will get this illness. 90% of them will be women. So what is so unique about this illness is that they actually have a very specific characteristic personality traits and behavior traits that is persistent throughout the course of illness, regardless of their body weight change. They are extremely afraid of high-calorie food. They want to consistently lose weight regardless of how thin they already are. And they're anxious, perfectionistic, and obsessive about food, even though they refuse to intake any. It is the highest death rate among all psychiatric illnesses. And what is scary about this disease is that they actually will will themselves to death. They know that they are sick, but they continue to not want to admit the problem and start eating and work with healthcare professionals. In fact, it took Rachel 10 years at the blink of death to finally allow herself to be checked into the medical center. And this is clearly a neuropsychiatric illness for that reason. People don't want to die from their illness, but patients with eating disorder like anorexia, they actually don't ignore the fact that they will die. So there's currently traditional treatment includes psychotherapy and some medication to treat depression. However, very little new treatment is being developed, even though we are discovering that there are neurocircuitry differences in patients compared to controls. The biological risk factors have been identified. They include the genetics, neurobiology, hormone, as well as the interactions of all these factors. So this is the most hot out of the oven review paper on the new treatment model for anorexia by world-famous Janet Treasurer of UK. Here you can see that with the predisposition risk factors, there comes the symptoms, and there's a vicious cycle of symptoms leading to secondary physical and emotional health problems. So what she recommends of the new treatment includes cognitive therapy and emotional support, DBP, to support the cognitive deficiency and emotional dysregulation. And then there is brain stimulation trial out there to assess whether or not they can reverse the neurocircuitry defect. And finally, what has been tried forever and hasn't seemed to work very well is refeeding and the eating behavior modulation. So with this, I want to just go back to my basic training. I'm a genetic epidemiologist. And just like many other complex polygenic disease, the genetic finding for anorexia nervosa has not been very good. It's not very promising. There's a lot of non-replications due to the sample size limitation, as well as the lack of homogeneous diagnosis criteria used by many groups. So what we took in the past few years has really helped in this regard in finding some otherwise unsuspected genes. In the first GWAS Association study in 2011, we took the top 1,000 hits of SNPs polymorphism, and we saw that 13 randomly distributed common SNPs of epoxide hydrolase 2 gene happens to be in the list. And with that in mind, it becomes a next investigation interesting candidate gene. So we then investigated a exon sequencing-based approach. We picked 150 genes such from the GWAS lead, and out of the sequenced exons of all these genes, EPHX2 surfaced as one of the few genes that were able to be replicated using the sequencing and rare variant cluster methods to be an associated gene with anorexia nervosa. That being said, how we found this gene was through rare variant clustering. And some common SNPs are indeed associated, but we would not have reached the genome-wide level of 10 to the minus of 8. So we then resolve this by replicate these associated common SNPs in multiple other cohorts, summing up to a total of 1,200 patients and about 1,900 controls. So the data I'm going to show you from this point on is actually a lipidomics study that's supported through this pilot grant, and hopefully the data can further convince all of you that this gene is indeed involved in anorexia nervosa. Oops. So the gene, it contains 19 exons, and many of the common non-SNPs have been associated with cardiovascular disease. And we are the first group to report that the gene is involved with psychiatric illnesses. The biological effects of soluble epoxide hydrolase has been well characterized by our collaborator at Davis, by Dr. Bruce Hammock and Christopher Maurice. Basically, the enzyme acts on the metabolite derivatives of polyunsaturated fatty acids. It turns the epoxy fatty acid into the diol fatty acid. A lot of molecular animal-based study has demonstrated that the epoxy fatty acid are protective anti-inflammatory metabolites that keeps the cells healthy. And when you have the epoxide hydrolase catalyze and loosen up this protective effect, it turns the host and the cellular system into a pro-inflammatory state. With that said, the Omics-Trio hypothesis surfaced. If you already know that the gene is involved somehow in the disease, and we know that the gene qualitatively and quantitatively change the protein, then it's very possible to envision, it's very possible to see how the change of this enzyme could lead to phenotypic changes. But if we were just to test this, we would be overlooking a lot of one other important factor, and that is the food intake. As we all know, since the patient have been refusing to eat, and when they do eat, they eat a very selective diet. This interaction between the food they intake and the enzyme perhaps is one of the factors that has a bigger effect to the disease onset outcome than just the gene alone. So to answer this question, in addition to the sequencing study, I conducted 4 small independent studies utilizing the metabolomics approach. We analyzed the PUFA in 60 patient total and 36 controls. We also look at a subset of them for the metabolomics. We call them oxylipins because they're derivatives of the PUFA. And we also took a longitudinal study design, well, semi-longitudinal. We basically look at the postprandial change of this metabolite to see whether or not the food factor is the food factor is important. Finally, we were able to measure the activity directly in the patient controls. So the fatty acid result shows that of all the individual omega-6 and omega-3 fatty acids, as you can see on the left here, the ones that were shown to be individually significantly different among patient and controls are enriched in the omega-3 class. We actually studied two groups of patients because patients ill, when they are ill, they have an average BMI in this particular study of 14. We then took recovered patients who have BMI of 21 and controls who are also at BMI mean of 21. And we also compare them because if this dysregulation or hypothesized dysregulation is due simply to the body weight change, then we couldn't pinpoint that to be an etiology factor for anorexia nervosa. And indeed, you can see that even though some of the dysregulated fatty acid normalizes to closer to that level of control in recovered patient, there still appears to be some abnormality to them. We then look at the ratio of such because the omega-3 and 6 have a very interesting competing relationship to the enzyme. We found that, again, the 6:3 ratio is consistently lower in patients compared to controls, perhaps due to the elevation of omega-3. But then we also identified that in omega-3 class ratios, there seems to be some evidence for the abnormality in omega-3 elongation and desaturation. The oxylipin, the metabolites we took and analyzed is a targeted approach. This is the assay design in Dr. Hammack's lab, and this is helpful to us because we already have a hypothesis that we are going after, and we are not aiming to discover new pathways for this particular study. Of all these metabolites, the ones in the blue fonts are the particular important metabolites that I included in my analysis. So compare the COX and 5-LOX, 12, 15-LOX, and Saturn 450 LOX pathway metabolites. The only ones that individually are different between patient and controls fall into the Saturn P450 one. Therefore, we can, at the current time, assume that Saturn 450 has a bigger role, plays a bigger role in etiology of anorexia than the other pathways. We also look at the ratio of the diol and epon and basically the diol fatty acid to epoxy fatty acid. This ratio is a marker of in vivo epoxide hydrolase level, and it's clearly showing that the evidence that anorexia nervosa patients have a higher level of such enzyme. Finally, we were able to measure the enzyme directly using the Buffy codes, and we were able to do this in the available recovered patient Buffy codes, and it's showing that indeed they are higher, even though the patient had been recovered for at least 1 year. So the newest pilot study, real quickly, I just want to show that I have then taken the changes of the metabolites, the changes before and after the meal. And we actually compared the diol metabolite in patient versus control. But in comparing so, I control for the variance of epoxy fatty acid and age, because I wanted to remove the substrate effect of the activity of epoxide hydrolase. And interestingly, I noticed there's a significant elevation of the 5-diheat in patient compared to controls. And using the same approach, I was not able to find any significant differences in all the diol fatty acid of the omega-3 classes. So this suggests that perhaps the effect of epoxide hydrolase activity is context-dependent, PUFA-dependent, and also the consequence of such interaction is driving to a pro-inflammatory state, which is leading to a neurobiological disease. So I don't have much time left, so I just want to summarize that we defined the gene, and it is being replicated over and over again to be an anorexic susceptibility gene. In the metabolomics study, we're able to identify the dysregulation that perhaps is associated with the dietary factor And using the postprandial readout, we are able to see that the food indeed plays a part in the metabolomic exchange, therefore suggesting a functional differences. In conclusion, both increased enzyme level and the dysregulation of fatty acid are involved in the pathogenesis, and perhaps through a pro-inflammatory cascade. And the dietary factor could modulate genetic susceptibility, like in many other diseases. And further study in using nutrigenomics or nutrigenetics approach is warranted. So I just want to show you again this previous treatment model recommendation. My goal is, and my hope, is that in the future we can use the screening of genetic profile as to identify increase of predisposition factor and also characterize the disease symptoms and the course using multi-domain biomarkers such as metabolomic strategy and output. With that could also become part of the predisposition factor predicting tool. And finally, perhaps the future treatment lies in finding out what the nutritional support is helpful in each of these stages of a treatment. Lastly, I just want to thank all my collaborators, the wonderful scientists at UC Davis, and the lipid mass help assay the fatty acid, and all the geneticists and eating disorder physicians at Price Foundation collaborator groups help contribute to the sample. Thank you.
Hi, are there any questions?
Do you think the sex difference is all societal or biological, and is it possible to get enough samples of men with this disease in order to start looking at that?
It has been difficult. It has been difficult to find just enough women alone to get a GWAS of sufficient power. So not many groups have really looked at the male anorexia alone. So, but that is something definitely worth looking into.
Thank you.
Thank you.